Okay, so how can we empirically test IIT-inspired measures of consciousness? So as I said in the last video, our brain is too complex and gigantic to actually compute the measure of integrated information. However, with a clever thinking and also tricks, various approximations can be made for the measure of the integrated information. And the question here in this video asked is that can the measure inspired by IIT be used to distinguish the presence or absence of consciousness in the brain lesion patients, patients under general anesthesia, and people in sleep? And if that's possible, then that will lend the support for IIT indirectly. So the key idea here is to stimulate the brain using a TMS, Transcranial magnetic stimulation, and then record the activation induced in as a sort of electroencephalogram, EEG, to estimate the integrated information. This is three examples of high integrated information with inhomogeneous connectivity, but still connected as a whole to make a big boundary. And another one is that highly connected and integrated, but doesn't have a much differentiation. And another one is that a disconnected system. And this phi estimate should reflect this to be the highest and this to be low and then low. That's a sort of clear prediction from IIT-inspired measure. So let's start with a situation where we stimulate this particular system, hypothetical system, with a TMS. And then if you, in this situation, the system is, doesn't have an integration, loss of integration situation. And therefore, it's expected to have a low integrated information value. And then if you stimulate this, unit number 1, because it's only connected to this, number 2, it can enhance each other or, propagate. But as the time goes from right to left, the initial activation should be, go down relatively quickly. And as a result, if you measure the EEG activity from each of these, 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8 systems, then 1 or 2 can be activated initially by this TMS stimulation, but 3, 4, 5, 6, 7, 8 remain silent. That's one prediction of this TMS EEG experiment applied to the system number 1 in a disconnected system. Then now, what happens if you look at the system that is connected too much and the inhomogeneous? And here, because of the homogeneity of the system, it doesn't have a differentiation, or otherwise, it lacks information. Here, stimulation propagates everywhere, really, strongly and uniformly, and then it gets activated initially. But because of the potentially, inhibition, excitation balance, and so on, it remains to it goes back to the baseline situation quickly, and then remains silent as the time goes from right to left, and that would correspond to this homogeneous activation, and then going back to the baseline quickly. And then finally, this kind of a complicated connectivity pattern, which attains a high level of integrated information. In this case, if you stimulate one part of the brain, then it can propagate to these areas and then activate this pattern, number 3 or number 5. And then each of the 3 and the 5 can activate this 5, and the 5 activates to this, and so on. And then that leads to this situation. And meanwhile, maybe 3 and the 5 can become silent without any further activation. But these two, 7 and 8, next activate 1 and also 3 and things like that, 1, 6, and then it propagates like that. So as a result, the prediction is that the activated pattern propagates across all the brains, connected brains, and then shows a complex pattern of the activity like this way. So these are the three predictions of a TMS EEG experiment when applied to the human brain with loss of integration, loss of information, loss of information, integration, loss of information, and high integrated information. Okay. The way the experiment goes is like this way. So first, the patient or subject will sit in a chair and then equipped with the high density EEG monitoring system, and that is compatible with the TMS stimulation. Okay. And then the TMS is held by this kind of mechanical device, and then it's connected to through this really thick wire to generate this massive magnetic pulse to the coil here. And then the location is controlled by some kind of system, neurobehavioral systems or something like that. And as a whole, it tries to generate an index called Perturbational Complexity Index, PCI, as I explain next. And this is the rough schematic of the principle of the transcranial magnetic stimulation very quickly. Basically, it tries to, it generates the magnetic pulse across this, circular coil, and that induces, the electrical current is passed through this, coil, and that generates a magnetic field indicated by this, red rings, and then that further induces the electrical activity in the brain, indicated by this green arrow. So that's how the magnetic pulse can be used to activate brain. So this is the hallmark experiment along this series of experiments performed by Marcello Massimini, currently in Italy. Initial paper was published in Science. And Tononi was also who was a proposer of IIT. I was directly, the person who was behind this experiment to design this experiment. And it's worth noting that before their experiment, TMS and EEG was considered to be possible to combine, unlike relatively easy, but it's technically also difficult compared to TMS and fMRI or EEG and fMRI. And even if you can combine this technique, if you don't have a right question, then there is no reason why you want to do this, basically. And here, the idea is to bypass any kind of sensory stimulation and motor output and directly stimulate the brain and then measure the complexity of the connectivity and activation pattern through stimulation evoked EEG. So here, the A part corresponds to the location of the TMS. Here is the anterior right side of the more above the frontal areas, and that's the location of the TMS. Okay, this is sagittal and also axial and coronal slice. And then red is, so this figure in B is showing each of the electrodes EEG recording from channel 1 to channel 60 or something like that along this y direction, and each of the diffraction corresponds to the diffraction of the EEG measurement. Okay, 200 microvolts here, and the x-axis is the time that goes from here to there. And then, as you can see, TMS pulse was applied in a red, vertical line, and each of the TMS pulse is separated by roughly like 2 seconds or so. And then during the wakefulness, if you do this experiment after stimulation, it doesn't look like nothing happens, and this is usually true for any kind of stimulation experiment which uses subthreshold stimulation. It doesn't evoke muscle artifacts or phosphine or something like that. Usually online, you should not expect strong activation induced by TMS, except for the artifact caused by the TMS, and they remove this artifact really skillfully. So you don't see much happening by the TMS in a single trial. On the other hand, non-REM sleep, where subjects are supposed to be not dreaming and in a deep sleep, the brain is actually highly active, and that's one of the things that I started off in this lecture. Activation, in terms of many different kind of definition, doesn't seem to be lower in any way when you lose conscious experience in the non-REM sleep. It's highly kind of active. And then after the stimulation of TMS, you seem to see something like coherently going activated here, and then around the same time it's also activated somewhat. It's a bit difficult to see still, but something is happening around the time of this kind of going down of the response around here as well. Okay, and it's very difficult to see on a single trial. So what they did is averaging across the trials, and then focusing on one particular electrode that is just under the TMS. Okay, one particular electrode under the TMS stimulation. Here's the average of smooth average of the electrical activity that is invoked by the TMS. Okay, so y-axis is the 450 trials that are transits from wakefulness to stage 1 and 1 sleep, and then this is an N2 sleep. And then up to here, each of the trial is noisy, but it's averaged over the 5 or 10 trials along this vertical direction. And then you start to see some kind of evoked pattern within this wake period, and that is clearly seen by this voltage plot, not the color plot in here. So this particular electrode seems to show that after the TMS, it goes up and then down, up, down, up, down, down, and then up and down, up, and so on. And this red line corresponds to the sort of the baseline activity, and above and below means that there is something significant happen. And then during the N1, at the onset of the sleep, where you might experience some kind of dream or so, or hallucination, there is some kind of activation, but it also goes down really quickly. And then in N2, where most people do not report seeing a dream, at least 50% of the dream, the sleep, there they found actually the activation by the TMS is actually larger initially, as you can see in this red part. So that is reflected in this large deflection here. But it also goes down and go back to the baseline quickly compared to the wakefulness. And this is consistent with what the theoretical prediction of the TMS EEG experiment was making about one particular electrode in the high integrated system. High integrated system should show something like this, and then low information system, homogeneous kind of system should show like this pattern. But to get to that kind of pattern of activation, you need to see all of the electrodes at the same time. And that's this next slide. So here, each of the figure component is complex, so I'll go through one by one. The first part here, the top row, is depicting the ERP averaged across many trials during the wakefulness. And each of the line corresponds to one particular electrode or channels. And there are 60 channels superimpose each other. And sometimes it's called the butterfly plots. So what it means is that, as in a previous figure, if you look at focus on particular electrode, just beneath the TMS, it goes up, down, up, down, up, down, 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 and so on. And then, so that corresponds to something like up, down, and then going up and then down kind of electrode. And then you need to superimpose all the electrodes trajectory, and then you see some of them go up, some of them go down, and so on. And then if you plot this magnitude as an height kind of plot, then you will see the second row of the figures. And here, the red one corresponds to either higher or lower activation, and then you can see it as this density of the line corresponds to the strength of the change over the time, over the space. And then each of the figure corresponds to a snapshot of each of the particular significantly modulated spatial pattern of the responses, and so on. And as you can see, what's important here is that the propagation of the activity goes, from right to the left and so on in a complex manner. And the last one is called current source density or source density estimate. Using some computational techniques, they were able to estimate how the activation spread based on this algorithm. And then here, this is 10 milliseconds after the stimulation. It is first activated. The neurons or neural structure beneath the TMS is activated first, and then that propagates to the front, to the left front, and then goes back to the back of the brain, and then come to the left again to come back to the initial part of the stimulation, and so on. And then at least it goes on and on up until 300 milliseconds or so. That's the wakefulness status of the TMS evoked EEG patterns, spatial temporal patterns. And then the next one is the pattern generated by TMS during the non-REM sleep. Again, non-REM sleep with most of the time, dream is not experienced. And here, initial activation is stronger than the wakefulness, as I mentioned. The magnitude is much, much bigger here. And that's I'm going to ask in the quiz. And then after that, it propagates a little bit, but it dies out also really quickly, and it doesn't move in terms of the spatial location. So this is corresponding to the situation of this too much integration and loss of the information or differentiation situation in the theoretical simulation, I mentioned above. And this is a sort of a schematic summary of the known result when this group, Massimini's group, has tested many various different kind of state of consciousness. And here's a schematic result. So here first, wakefulness, if you stimulate this particular area of the brain by the TMS, then it propagates to other areas and also sustains activation for a longer time. And during the non-REM sleep, sometimes weaker stimulation, like 90 volts per meter, that shows activation only one pattern and then one location, and it doesn't propagate. If it's too strong, then it goes to many areas at the same time and then propagates, but it dies out as well quickly. Okay, so then what happens when they did the experiment when people are dreaming in a REM sleep state? So it's almost like in a wakefulness situation. A little bit of the stimulation in one area propagated to many other areas, and then it doesn't come back to the baseline for a longer time. And under the anesthesia, where researchers know that people lose consciousness, then it's similar to the situation of the non-REM sleep. Stimulation by 120 volts per meter, doesn't propagate much and then dies out quickly. And then, most critically for the clinical purpose, vegetative state or unresponsible wakefulness states, these are the people who do not seem to have consciousness in many criterion. They have similar type of local disconnected type TMS response, like anesthesia or non-REM sleep in this situation. And then minimally conscious and locked in state patients, they show much more reliable signals of consciousness. And the lock-in syndrome is the one that I showed the video in the earlier videos, and they are fully conscious, but just cannot move. And then they are almost like wakeful subjects. Minimally conscious patients who fluctuates in conscious experience or conscious states, but when they are tested with the conscious state, they show similarly complex pattern of the TMS evoked EEG patterns. So as an interim summary, TMS evoked EEG patterns were expected to differ depending on the varying states. High integrated information system should generate more complex TMS EEG pattern, and the low integrated information system should generate less complex TMS EEG, either activated all the time or activated only parts of it. And then both of them were confirmed across different types of loss of consciousness. And then the next question would be, can we construct a consciousness meter based on this idea? And that's what I'm going to talk in next video.